Hello students, again I am here with marine and freshwater microbiology, and today we have to talk shrimp bacterial disease. In marine and freshwater, it is a huge source for the business of the human being, as well as it have a big source of the food for the human in terms of fisheries as well as in terms of shrimps and other water related businesses. If water is fresh, the living organism, fish and other marine organisms also will be healthy. If marine water or fresh water is contaminated and it have various type of bacteria, viruses and fungi, it will cause disease in habitants of that environment. And habitats of in the marine and freshwater environment are fishes, shrimps, and prawns, and many more organisms are there which are contributing in human economy as well as in human food and diet. Therefore, it is very important that when we are going to study marine and freshwater microbiology, we have to keep in consideration that freshwater or polluted water also affecting the life of other living organisms in marine and freshwater. Consequently, which is loss, causing a huge loss to the relevant businesses, as well as if contaminated fish will be supplied in market that will enter into the food chain and consumers also will suffer in various and different diseases. Therefore, it is important that water must be free from pathogenic microorganisms. So here we will talk some shrimp bacterial disease. Shrimp bacterial disease covered Vibriosis, necrotizing hepatopancreatitis, hypocommensal falling disease. These three categories of disease are has been found in shrimps. Shrimps vibriosis, known in Latin America as the seagull syndrome, due to shrimp swimming at surface of pond as seagulls eat them. So this is one scenario known in Latin America as seagull syndrome due to shrimp swimming at surface of the pond. Seagulls eat them. Numerous etiological agents: Vibriosis harvoi, Vibriosis valencificus, Vibriosis parahemolyticus, Vibriosis alginolyticus. These are the etiological agents of the Vibriosis. Wide variety of gram-negative motile rods also causing infection in shrimps, most frequently found in hatcheries, but a big problem for young pills in pond. So this is very sensitive region, that is hatcheries where fisheries are small, neonate and very fragile to the different infections. If some bacteria or pathogen will enter in hatcheries, that is causing huge losses in fishes. All shrimp reared under stressful conditions are susceptible. Shrimp vibriosis. Clinical signs of the shrimp vibriosis are high mortalities, fish will start to die and it will not stop, and place young juveniles, moribund shrimp appear hypoxic and often come to the pond surface or edge. Seabirds preying on shrimp. Presence of the luminescence intense. So whenever the shrimps they are getting infection due to the pathogenic microorganism or viruses, they are coming on the surface of the pond or lake or as well as hatcheries. and they become a prey of the birds and other animals from that region. Presumptive diagnosis, clinical signs, large amount of bacteria in hemolymph, slow clotting, melanosis of shell, confirmatory diagnosis, isolation and purification with appropriate media, which can be used in laboratory, that we have to collect some specific samples from uh, pond, and either we can collect dead shrimps, or we can collect sick or ill shrimps, and we can isolate the infectious agent from those shrimps by using selective or appropriate media, as we are API redundant NFT strips or TCBS media. Shrimp vibriosis commonly affect organs. It affects cuticles, hepatopancreas, midgut infection, lymphoid organ, antennal gland, heart and hemolymph striated muscles. These are the different sites or organs which are affected due to uh, vibriosis in shrimps. Shrimps vibriosis, hepatopancreas, if you will see, most form in Central America. Evaluate shrimps on weekly basis for vibriosis. So mostly they have facilities and they are active. They are getting too much revenue from this business. So they are, if someone have a routine business of it, they are doing this vibriosis basis for vibriosis. They are checking fishes weekly for vibriosis. Gross signs, blast spots and cuticles will appear. Internal signs, evaluation of the hepatopancreas using wet squash and evaluated blind tubules for the construction presence of G rods. Alright, hepatopancreas on a scale of 0 to 3, 3 being worse, 3 is worse. Medicated feed at 4 gram per kg oxytocin at nitrofurazolidone, nitrofurazolidone and serafloxacins, which is called serafin. And this type of medicated food can be used to prevent the fish shrimp from disease. Hatchery control, improve husbandry, especially in the area of the sanitation, quality feed, water source must be free from contamination, water source purity, use of probiotics and vaccinations in terms of seraphine or antibiotics we can use to save shrimp from vibriosis. Grow out control, improve stocking. Grow out control, whenever we are growing in a controlled environment, we must have to keep this thing in control, improve stocking, handling to reduced stress, have feed in pond in advance of stocking, use of molasses and nitrates as fertilizers. Such like signs will appear, black spots as well as these things will appear on shrimp skin or on body. So we are going for the next, that is the necrotizing hepatopancreatitis, also known as NHP or Texas Pond Mortality Syndrome for Obvious reason, this is a disease of the midgut gland, not as with a vibriosis, the blood. Bacterium prefers high salinities and it forms at more than 10 ppt, and those bacteria which are found at high salinity environment means in salty environment, uh, they can infect to the fish and they are causing necrotizing hepatopancreatitis. Believed to be an agent of this causative agent of this disease is believed to be a new genus of the Protobacteria alpha group, that people are considering that a member or a group of the Protobacteria causing this infection in shrimps. Found from Peru to Texas, this region is most common where necrotizing hepatopancreatitis found, and that region has been studied, might be found in other regions of this globe or ocean, but not has been studied yet. Small G-negative exist, gram-negative bacteria exist in two morphological forms, rod-shaped rickettsial-like and flagellated helix. Gram-negative bacteria exist in two morphological forms, rod-shaped rickettsial-like and flagellated helix. These are causing the necrotizing hepatopancreatitis in shrimps, and mostly this disease has been studied in Peru to Texas. And these type of bacteria which are gram-negative rod and rickettsial-like structure, these are found at high salinity uh, areas, mostly in we can say marine water. Necrotizing hepatopancreatitis host range are, this one is P. venomine, P. asiaticus, P. certiferus, and P. stradrotus, and P. californiensis. These organisms are most causative agents which are causing hepatopancreatitis in shrimps. Diagnostic method: presence of massive number of the gram-negative bacteria in hepatopancreatic tubules epithelial cells. First sign will be this, that they contain the most high number of the microorganisms. Epithelial atrophy of HP pelvis, HP collar, DIG level DNA probe using in situ or blood hybridization, and the transelectron microscopy of the hepatopancreatic cells showing granulomatous lesions. So by this way we can diagnose hepatopancreatitis in shrimps. We can use we can we can count the gram-negative bacteria from that epithelial cells of shrimp. Atrophy of failed HP collar. We can do level DNA probe using in situ and don't blood hybridization team. Different techniques we can perform for the diagnosis of this disease in shrimps. Clinical signs: reduced feed intake. What will be in fishes? They will reduce intake food, empty gut, anorexia, poor ratio filled period hepatopancreatitis. Simple diagnosis: most farmers use wet mount of HP and look for reduced lipid droplets myelinization of the tubules. So most commonly by a traditional method, this technique is used. Most farmers use wet mount of the hepatopancreatic pancreatitis and look for reduced lipid droplets, myelinization of tubules. Control, so how we can control this thing? Frequent histopathological examination, use of the oxytate at 4 gram per kg, 4000 ppm, avoidance of the high salinity condition. So this medicine can be used for the control of this disease, hepatopancreatitis, in controlled aquaculture systems like this. Necrotizing hepatopancreatitis is like this. Zoea II Syndrome, problem facing hatcheries through the Western Hemisphere. It is a problem of the hatcheries and mostly in Western Hemisphere. Condition results in heavy mortalities, mainly concentrated in the Z2 substage of the larval penial shrimp. Syndromes are a group of signs that occur together and characterize a particular abnormality. First characterized in the paper by the Lornizo Jets group in Florida. Zoea II syndrome, because different larval disease can share common clinical signs, it is difficult to characterize a distinct pathological agent. Could be associated with water quality, nutrient, or pathology. So that syndrome may be associated with water quality. Again, if water quality is good, there will be no Zoea syndrome will be in fishes. If nutrients are good and pure from pure, that shrimps can be saved from the Zoea syndrome or pathology. It is felt to be a distinct syndrome because of its life stage specificity, a remarkable similitude of clinical signs reported throughout the America, not noticed prior to 1993. So it seems that after 1993 there is huge contamination in water bodies which caused infection in fish and in shrimps. Species affected P1ME and P. stylirotus, although primarily the former. Clinical signs normally appear normal and healthy metamorphosis to Z1 and start eating normally. Long facial stands are exhibited. 36 to 48 hours after achieving Z1, first sign appears: anoxia, evocation of gut, lethargy, erratic swimming, loss of normal pigmentation. So these are signs of the Zoea II syndrome. Death due to starvation in Z1 to 2 moles. Mortality stopped at Z3. Survivors continue normal larval development. Internal pathology: atrophy of the digestive gland, inflammation of gut walls. Possible etiologies: water toxicity, bacterial pathogens, viral pathogens, no response to antibiotics. 1997 agent determined to be intracellular bacteria found by team in hepatopancreatitis. As of yet, no known treatment. So after this, we have discussed the disease, shrimp bacterial disease. So marine water or fresh water. Uh, these have a huge source for the human uh, economy as well as human food. If water quality of marine, ocean or fresh water is good, the living organisms of which are growing over there, such as fish, shrimps, and other marine organisms, which have huge contribution in human economy as well as in human and animal diet, their health will be good because there are many people who have business with marine with aquatic life, and they are contributing huge GDP to their national economy. If water quality is good, they will the disease rate and the death rate of these organisms in either they have been grown in controlled environment, either has been grown in natural environment. The mortality, death and loss ratio will be reduced if water quality is good. If water quality is not good, what will be the people who are involved in such business with aquatic organisms? They will get huge loss, and as a result, they will suffer, and it will affect the human economy throughout globe because marine organisms are transported from country to country, and one suppose those oceans which have no reservoir or no ocean, they are getting marine organisms from other countries which have coastal region or oceans, and a huge amount of food in terms of fish and in terms of shrimps and other organisms are taken from the oceans. So we can assume from here that water quality is very important. If water quality is not good in any way, either in direct consumption of the human, it will affect the human health. Either we are growing some animals in that water for the agriculture, for business purpose, or domestic purpose, or commercial purpose, we can face huge losses if water quality is not good. So we must have to keep water free from all type of microorganisms, especially pathogens. So water is a basic requirement of the every living organism on the globe, and it is quality can and it is quality and it is hygiene condition can make life easier and good of the every living organism on this globe. If it is quality is compromised, it will simultaneously or consequently it will affect the health of the human and other organisms. So for detail, you can go through our book and you can discuss with your colleagues and friends, as well as you can see some research papers. I hope you will try to understand as you have to go through our book to discuss this thing in detail. So thank you very much. Have a nice time.